Hello and welcome to today's webinar hosted in partnership with Oxford Biomedica. I'm James Miskin, the editor at Facilitate, and today I'm joined by a great panel who will shortly be introduced to you. But before I hand you over to today's moderator, I just have a couple of housekeeping items for you. First of all, I would just like to remind you that the majority of this session is reserved for an interactive Q&A discussion. So please do submit your questions via the Q&A widget available at the bottom of your screen. We will also attempt to answer your questions offline should we run out of time. This session is being recorded and will be available on demand shortly after the conclusion of the event. Details for on-demand viewing will be sent to you once the recording is available. So without further ado, I would like to introduce you to Jessica Carmen, James Miskin and Sandy Douglas, and I will now hand over to Jessica to start today's discussion.
Hi, everybody. Thank you for joining us today. We have a very lively discussion planned to talk about the manufacture of COVID vaccine that was developed at University of Oxford and is manufactured in part by Oxford Biomedica. We're going to start with, Sandy's going to start to introduce the collaboration. Sandy.
Hello, everybody. So I'm a research group leader at the University of Oxford. I've led the university's work on manufacturing scale-up for our COVID-19 vaccine. And just to set the scene for this discussion, a few slides about our team in Oxford, the platform technology, a little bit about the manufacturing process, and then in very high level overview, the story of how we came to be working with Oxford Biomedica and AstraZeneca. So pre-pandemic, there were about 200 people working in Oxford on vaccine development, spread across a number of different groups with slightly different flavors of interest. And I won't list them all, but the key point is that there's a wide range of different capabilities and specialist interests represented here, which came together as a team of 6 of us principal investigators working on different aspects of the program. So Sarah Gilbert, vector development, Adrian Hill, a lot on the commercialization strategy and the original vision for the Institute, myself on the large scale manufacturing, Tess Lamb, a lot of the immunology and vector development, Andy Pollard, clinical trials, and Kath Green on the pilot scale GMP. So it's been a real team effort, each of us leading teams. On to the next slide, please, Georgie. Thanks. So the reason we were able to move fast was the combination of that pre-existing capacity with a suitable technology. So that's the ChAdOx1 viral vector, adenovirus vector platform. And in particular, through work done by Sarah's group, this had been applied pre-pandemic to another coronavirus, MERS, and Sarah and Tess had shown in primate challenge studies that it was protective against MERS, and had got that into a phase 1 clinic. So we had pretty good confidence that this vector platform was suitable in principle for a vaccine against this novel coronavirus. Next slide, please, Georgie. So in brief overview, the manufacturing process itself, what we have now, and I'll tell you on the next slide how we got here, but the process is not that different. Some people are surprised how similar it is to processes for manufacturing other viral vectors or indeed recombinant proteins. So HEC293 T-Rex, which is an antigen repressing cell line, expanded in suspension in stirred tank reactors. That's then infected with an adenovirus seed virus. The antigen in that is under a repressible promoter. That's important because it means that the platform behaves the same way, whatever antigen you're targeting. The cells are then infected, and it's a fed batch process lasting a few days, and then harvest. The cells are lysed, nuclease treated, depth filter clarification, and then on into a downstream consisting of anion exchange and TFF. Next slide, please. So this slide's rather text dense, and again, I'm not going to ask you to read it all, but really the reason I've put it in set out in this way is just to try and give a little bit of a flavor of the tempo with which things were moving early in 2020. So before February 2020, the university had made small batches of many different adenoviruses, but never more than a few thousand doses of any one thing. And we'd never gone above a few liters. In February 2020, as we became alarmed by the new coronavirus, we decided to scale aggressively from an initial observation by a postdoc in my lab, Karina Joe, who'd found with incredibly lucky timing that she could increase the output of our previous process about 5-fold with a change of medium and the feed. We scaled from that initially to 3 liters in-house, and in parallel with that, we formed a consortium by reaching out to a range of potential collaborators and asking for help. We knew we needed help, and the first group of collaborators on board were Paul Biotech, Cobra Biologics, Helix, so that's 2 CMOs, one in the UK, one in the Netherlands, and the Vaccine Manufacturing Innovation Centre. Together with them, we were able to get a small rapid response grant from the UK government with what seemed like a pretty preposterous idea. Or, I hesitated to write it because I thought people might not think it was credible that we might be able to make 1 million doses before the end of the year. And the goal in that grant was a 200-litre engineering run and preparation for tech transfer. We weren't actually seeking to fund, actually do the production of 1 million doses. It was just to try and make it possible. March 2020, that plan got put into action. We went to 50 litres in Paul's lab near us in the south of England. And then 3 further partners entered our consortium. So Oxford Biomedica, who you're going to hear much more from James, and have been key partners in the UK, Wuxi Biologics in China, and the Serum Institute of India. So within March, we had tech transfer underway to 5 sites in 4 countries. We had something like an 8-way CDA arrangement. And we still had very little money. Everything that was going on beyond that award of £400,000 from UK Research and Innovation was being done at financial risk. So we were having lots of conversations saying, well, someone's going to have to put in some money at some point. This makes sense, but we need to get on with things now. And we were incredibly grateful to all of our partners who were willing to sign up for that. April 2020, it was when it really began to feel as though the cavalry might be arriving. And what happened then, we got our 200-litre PD run done at Paul. So we felt by that point we had proof of concept that this product could be made at meaningful scale. And previously, we'd been really quite worried about whether any serious partner would, big pharma partner would be willing to work with us because we had no demonstration we could make the product. We had our first GMP work happening for the large-scale manufacture in April. That was our working cell bank, and we got £65 million committed from the UK government. May 2020, more going on in the clean rooms, tech transfer happening at pace, and as people know, AZ joined the partnership with us at that point. And then June 2020, 200-litre GMP batch and a 1,000-litre PD run at AZ. So things moved really very, very fast. And to be frank, we were quite fortunate that things went really better than we could have reasonably dreamed to hope. Next slide, please, Georgie. And where we've ended up with input from a large number of partners, Oxford Biomedica and many others, under the oversight of AstraZeneca, has been a distributed manufacturing platform producing drug substance, I think, in about 12 different countries around the world now, drug product in several others. In short, a large number of parallel regional supply chains. This has now produced more than 2 billion doses of vaccine. It's important to emphasize, I think, the fact that although we've had these distributed manufacturing hubs. It has all been under a central umbrella, which has ensured coordination between the two and consistent coordination between the many and consistent quality. That's been a really very fast run through things. I hope it's given enough of a flavour of what happened from our perspective to set the scene for James and then for some discussion to follow.
Thanks very much, Sandy. So if we can move on to the next slide. So very quickly, for those of you who don't know, Oxford Biomedica, so we are located around about 1.5 miles away from where Sandy's based in the Jenner Institute in the University of Oxford, in Oxford. We're a publicly listed company. We've been on and we're traded in the London Stock Exchange, and we've been on the full London Stock Exchange since 2001. We've got over 740 employees, primarily in the UK, and a small number in the US. We also have an office in Dublin, specifically for batch certification activities and release. And as a company, we've built a strong reputation over the 20+ years that we've been in existence as a leading company in cell and gene therapy, primarily focused on viral vector manufacture in and around our proprietary lentiviral vector technology. So clearly from that, you can see we were not a company with a strong heritage in adenoviral work before this. We'd done some research in adeno, but we'd never manufactured a batch. Next slide, please. So obviously, as Sandy said, we started talking to the Jenner around about that same time in February. And as a listed company, it took us a little while to get full alignment, first of all, to understand what it was that we were going to contribute to this consortium effort, such that we would be able to, let our shareholders know what we were going to be doing. So in joining the consortium in May formally, we actually started working in April. We were able to really start contributing to the consortium effort. And to Sandy's point, we were working as one of many organizations collectively, all with the same goal. And these were organizations that, historically might not necessarily have naturally all worked together on one project. So this was quite new. Starting early on, we were considering manufacturing at 200 litre scale, and the experience we had as a commercial viral vector manufacturer was sort of helping to build the case. I think once AZ joined a little bit later, the good thing, at least from my perspective, was that AstraZeneca decided not to start again from scratch. They basically decided to adopt the philosophy of what the consortium had worked up at that stage, and basically endorse it and accelerate it even further. That sort of gave us some real confidence, and we started seriously considering scaling up beyond 200 litres at that early point. We, through the course of those next following few months, we were initially in discussions with the UK Vaccines Manufacturing Innovation Centre, ultimately signing a partnership, which we'd always intended to sign as an organisation local to us. And we'd started talking to the leadership at VMIC about the potential for collaboration, not about COVID. Then COVID came. The decision to collaborate then became quite an interesting one, because like many other organizations in the early phases of the pandemic, we didn't really know what it meant for us as a business from a financial security point of view, and consequently to invest in a very uncertain project at financial risk was a challenge for us. We did do that, but it was very helpful that the VMIC agreement allowed basically the use of government funding to purchase some of our key equipment that we would need for the various manufacturing suites. I probably should have started this to say that we had very serendipitously just opened our new Oxford manufacturing facility called Oxbox. And just on the right hand side, you can see a picture of the UK Prime Minister Boris Johnson at the opening ceremony in January last year. So we had intended to bring that facility, which contains 4 fully independent viral vector suites, up and running over the course of 2 years through 20 and 21. And essentially what we decided to do very early in the consortium was to accelerate that. And what that meant as a business is bringing a lot of people and an enormous amount of new, not foreseen equipment into 3 of our 4 vector manufacturing suites. So fast forward to month 9 post start, and we're initiating manufacturing of production at 1,000 litre scale by this stage of the vaccine in 3 independent lines, and we're from that point onwards manufacturing flat out, back to back. And obviously, history has said that the vaccine was approved by UK authorities in December 2020, and materials started going to supply at that point. So for us, this was a tremendously exciting opportunity, as it was definitely the right thing to do from a company perspective. It was definitely the right thing to do from a personal perspective, and I think we're just here today to sort of give the sort of commercial view to this collaborative project that the University of Oxford really kicked off from day one. So I think with that, I guess it's obvious that we've made tens of millions of doses of vaccine, and that's contributed significantly to that total of over 2 billion now at this point. So I think that's the last slide. Move on one more, I think. Yeah.
Yeah, I believe that's the last slide. I want to remind all of our participants that they can submit questions using the Q&A widget at the bottom of your screen. If you enter the questions, they'll come to me and I'll ask them of our panelists if I'm able. We're going to be focusing the discussion today on CMC and manufacture of the vaccine only. I want to just remind everybody that we're not going to be talking about any clinical aspects of the vaccine here. So while we're waiting for some questions to come in, how about if we start out with how did this happen? You know, who picked up the proverbial or not proverbial phone and said, hey, I have a great idea?
So this began to happen in early February 2020, and there was an initial phase of, for myself at least, increasing anxiety and thinking, well, what on earth are we going to do about this? You know, as a university, we have no experience of producing anything on a scale that anyone would think was commercially relevant, let alone enough to deal with a pandemic. Sure. And my training's in clinical medicine. And one of the things you get trained in clinical medicine is that when you encounter a serious emergency situation, you don't try and deal with it yourself. You call for help first. You know, anyone who's been on a resuscitation course probably knows that. And so we called for help. And there were two key things there. First, I picked up the phone to a guy called John Humphreys, who was head of process development at the Vaccine Manufacturing Innovation Centre. At that point, the Vaccine Manufacturing Innovation Centre was six people in an office on the Oxford Science Park. There was no innovation centre. It was a plan on paper. But John did a fantastic job in making further connections for us. And secondly, my colleague Kath Green, who runs Oxford's GMP pilot facility, sent out an email to the UK Bioindustry Association mailing list saying Oxford's doing this and they want help. They'd like equipment, they'd like advice on tech transfer, you know, can anyone offer anything? And the replies to that came in really thick and fast from all over the UK bioprocess community. And I think it's a fantastic, you know, tangible example of the importance of having networks like that to promote competition, because we wouldn't have got off the ground without that.
The ask and ye shall receive. It's always good to ask for help, and leveraging local networks certainly makes a lot of sense. It's pretty great. So some questions are starting to come in, which is fantastic. I'll read those in just a second. So I think it would also be valuable if we talk about maybe why was ChAdOx the ideal platform for this vaccine, and how did the Jenner Institute leverage the experience they already had with the platform in order to quickly retool it for SARS-CoV-2?
So adenovirus vectors, we had done phase 1 and 2 clinical trials across the different vaccine groups in Oxford, probably with maybe 10 or a dozen or so different chimpanzee adenovirus vector vaccines against different diseases. And before the pandemic, this is. And we knew across all of those that the safety profile was really very consistent. The reactogenicity is a property of the vector, not of the antigen it encodes. And also the immunogenicity was consistent. We knew, whatever antigen we put in it, we're going to get an antibody response. We're going to get a robust T cell response, including cytotoxic CD8 T cells, which is important for viruses. And we knew, as I said, that this had proven protective against MERS, another coronavirus in primates. So with all that, it's technically, having done that groundwork, it's relatively straightforward to copy and paste a new antigen expression cassette into the vector, and then turn the handle and do what we'd done before. The difference, obviously, was the speed with which we turned the handle and how far things progressed. In terms of what has made it a useful platform later, once it reached the more advanced clinical trials and deployment, it's been shown now that it is possible to manufacture these vectors on enormous scale. The manufacturing process was, it was new. We hadn't done this at even 3 liter scale, exactly this process at the pandemic, but it's based upon a skeleton of unit operations which are familiar to CMOs all around the world. The vector itself is BSL-1 in some jurisdictions, BSL-2 in others, so not a big containment challenge. Quite productive in terms of output per unit volume, and it's robust. And it's a non-envelope double-stranded DNA virus. So for people who are used to working with lentiviruses, for example, this thing's pretty. You can get it wrong, but it will tolerate quite a bit. More
forgiving, maybe.
Both in processing and in distribution, and that robustness in distribution has been really important in terms of the global reach of the product and making this complementary to the mRNA vaccines, which have obviously been fantastic, but less suitable for getting to some parts of the world.
Okay, that's awesome. So we do have a question that's come in already. And as a reminder, please use the Q&A widget at the bottom for everyone who's watching. When switching to 1,000-liter manufacturing, what was the validated scale-down model? In another word, do you still rely on process development bioreactors such as Amber, or you had to use larger volumes such as pilot runs? James, I believe this is for you.
Yep, yeah, I can take that. So in essence, as Sandy said, when the process first started through the consortium, it went up through lab scale, through 50, 200 litre scale. There were some challenges to the process in terms of the unit operations. There were some Herculean volume flow rates and challenges that we as an organisation, amongst others in the consortium, were concerned about. And actually, one of the things that we needed to do was sort of refine the process parameters and change the process a little bit from the process that was originally developed by Sandy's team. And to do that, you need to do that in small scale. So ourselves and other organisations, including AZ themselves, did quite a lot of work at lab scale. We did work at both the 5 litre and the Amber 250 scale to further sort of refine, if you like, the upstream process. Obviously, for downstream, we needed to have a reasonable level of volume to be able to perform the downstream in a scale manner. So most of that was done at the 5 litre scale. But yeah, it's absolutely like any scale up process. It's also important to have a relevant and ideally validated scale down model. At that stage, the scale down model wasn't validated. As Sandy said, this was a project that was moving at a tremendous pace. And so what we had is a timeline to get us to the first commercial manufacturing, to be in a position to use that initial material. The first material that was made was for clinic, but the plan was very soon to get into material that was made by the final commercial process, such that if it's approved and if the data comes in successful from the trial, then that material will be suitable for commercial use. And I think it was really, really important to be able to apply the sort of process knowledges in terms of unit operations, as Sandy's mentioned, but also understanding how to validate scale down models. And again, the work was split across a few different organizations to make sure that we could cover off all the different parameters that we needed to.
Actually, a question that comes to my mind is, now that that work has been done, if there were to be another coronavirus or other disease, non-corona, but something else, Ebola or something, would it be even faster to use ChatOx to address it because this work has been done?
Or a variant, perhaps, God forbid. The short answer is yes, it would be substantially quicker to do this again tomorrow. The people may have heard about the 100-day challenge from CEPI. The idea that you could go from identification of a new pathogen to release a vaccine in 100 days. That includes multiple aspects, trials as well as CMC. Mm-hmm. We're not quite there yet on adenovirus vectors in terms of this commercial CMC within 100 days, but we're not as far off it as people think. There's a fairly widespread perception that adenovirus is slow, mRNA is fast, adenovirus is slow. I really don't think that's true. There was a market analyst was quoted asked by a journalist about would adenovirus be fast in response to Omicron, and he said adenovirus is offended by being asked to scale up quickly. It takes it personally, which is total absolute nonsense. I think mRNA does have an edge by a couple of weeks, but really it's only a couple of weeks. We could do this much faster.
I think just to add to that. Go ahead, James. Yeah, just to add to that, I think, clearly to get started in any adeno process, you need things like a cell bank, but you could have that already in place. So that was on critical path in the original project. It wouldn't be the second time around, assuming the same platform is used. So essentially all you need, in inverted commas, if you're sticking with the platform approach, is your viral seed. And that viral seed, the difficult bit is the cloning, is the changing the antigen in that vector backbone. Once that's done and you've grown up your stock, then you're ready to start manufacturing. So from that perspective, that isn't that dissimilar to those of you who are cell and molecular biologists from the challenges of cloning into a plasmid and starting to generate sufficient plasmid to generate RNA template. So it's, yes, it probably is a bit slower, but I think this kind of insistence to sort of make one platform better by making another platform look worse, I don't buy. I think there are plenty of advantages to these kind of technologies. Sandy's already talked about one obvious one, which is, the shelf life. But in saying that, you don't need to, there's no debate about the value of the mRNA platform technologies because they've clearly proven their worth as well. And I think it's been vital to have multiple shots on goal here, because at the start of this, nobody knew whether any of these platform technologies were going to generate a vaccine that would be efficacious. And I think what history has now shown is that many of them have, and that's great.
No, that makes a lot of sense. So sort of to finally dispel this myth, that if a variant were to come up where new vaccines were required, there's no reason to believe that adenovirus vaccine would be significantly behind RNA.
I personally don't think so, no.
Okay.
I think actually the challenge, the rate limiting thing probably is actually the trials rather than the CMC. As long as investment is made to get on with the CMC at financial risk ahead of the trial results, there's not a meaningful difference in the timelines, in my view.
Yeah. And that's the common, the financial risk is the key point. Ultimately, when this project was started and we, Sandy and team were able to secure that UK funding, I think that UK funding basically underpinned the ability to go forward at pace. Nothing was, no sort of safety risk was employed. It was purely you're making a vector that could be a vaccine in the future, and if the efficacy data doesn't support it, then you've basically just spent a lot of money making stuff that isn't going to work. So that's the risk. It wasn't a risk in relation to anything else. And again, that would be the implication there is if there is a willing developer to take that level of financial risk, then speed can be of the essence.
It was quite interesting in the discussions I had about that investment at risk, really at risk. The UK government had more or less committed to give us £65 million before the first patient had been injected in the first clinical trial. In the discussions I had about that, people in science, people in pharma really balked at it initially. They might not today, but they did then in many cases. The people who got it were people from a more sort of economics or financial background. They were quite happy with the idea. If there's a 10% chance that this is going to work and the value it delivers is going to be more than 10 times 65 million quid, well, let's get on with it.
It struck me when you were going through the slides in the introduction, the incredible luck, of all the pieces sort of falling into place. But I think a lot of it too, the fact that the money was there due to the global impact, and sort of what we were saying, right? The investment had to be there because something had to be done. And it reminds me of that adage, of life in general and manufacturing in particular. You have the options of fast, cheap, good, choose two. Right. So it had to be fast and it had to be good. So, the money had to come.
Yeah.
Yeah, I think that's a really interesting point when reflecting on, well, what is the learning from this that is transferable to other projects? I think a lot of people look at this and they think, oh, well, brilliant. We've learned loads. We're going to develop all the drugs of the future much, much faster. Well, actually, unfortunately, many of the things that did work in this worked because of a unique strength of business case. And it wouldn't make sense to make quite the same financial risks for virtually anything else. So I think in trying to draw out which things we did you actually could replicate, one needs to think really quite critically about which aspects, won't hold up.
Yeah, that's true. Yeah, and let's hope we're done with global pandemics for our lifetime again, at least. A question came in that's directed for James. Do you think the lentivector platform is the future or an alternative technology? I don't know if the question is related specifically to COVID vaccine or just pharmaceutical cell and gene medicine. So James, take it as you wish.
Sorry, Jessica, just before James comes to that, I just wondered maybe if I could say a little bit more on that previous point about financial risk, maybe ask James for his thoughts on it as well. Because, some of the financial risks that we took were replicable. And it was about trying to be really quite scientific and clear about which risks were patient safety risks that we weren't going to take and which risks were actually a business risk. So, for example, transferring a starting material from one facility to another and accepting it in without GMP validated, mycoplasma and sterility results, but maybe with a, preliminary bioburden. Well, that's a business risk. The result's going to be known long before the product goes anywhere near a patient. And the probability of anything going wrong, if it's come from another GMP facility, is very low. So we took lots and lots of informed risks like that had no patient safety impact, not usual practice in certainly some parts of the industry. But I think actually that could be done again if people have the guts and maybe the insurance.
Yeah, it's the appetite, I think. I think it, and basically liabilities. If you're a CDMO manufacturing somebody's crown jewels, and you might gain an extra week by not waiting for some of those crucial data points, and materials are in short supply, then you're not going to take that risk. But in this case, it was obvious that absolutely you'll find out very, very quickly if that was a risk that you shouldn't have taken, and then the batch won't be successful, and you'll have to wait for the next one. So from that perspective, those are precisely doable again on the proviso that the organization who's taking the risk, or the organizations in this case, because it was a multiple organizations making decisions, would accept that level of risk. And I think that's the point you were alluding to earlier, Sandy. For other non-pandemic drivers, where there isn't that sort of urgency to an extent, these are very important things to address, but you're trying to develop a product that might take many years to get to the market, then it's not going to make the same degree of sense to take those sort of more risky business risk decisions, because ultimately that just hits your budget.
I absolutely agree with you, but some of them are sufficiently small that I think it will be rational to take, and part of it comes down to sort of people's, you know, legal positions and contractual positions. You know, so I had. There were certain conversations I had like a stuck record. I was having the same conversation with people again and again, and one of them was, I would like you to tell me how quickly you might be able to do this if everything goes to plan. I don't want you to tell me how quickly you can guarantee you can do it. I don't want you to tell me how quickly you would sign up to a contract to do it with someone who is going to hold you to it. I want your honest best estimate of something that's realistic, but if things go well. And I don't know how you translate that way of doing business into the usual world, because all of these early conversations, there were no contracts. There was just a promise that somewhere down the line there might be one. And that huge value came out of that, because people were talking very openly, genuinely collaboratively. Everyone's interests were aligned. What can we make possible?
No, that's very true.
Yeah. I think it was important what you said too, of the recognizing the business risks that would still in no way put patients at risk. But right, so the business risk is you have to run another batch, which is a money sink, but there's never going to be a risk to patients. And that's, you're right, you know, with an indication that addresses less than the global market, you know, maybe that's not a risk worth taking, but in this situation, it certainly was.
But I mean, just to maybe challenge that slightly, loss of a batch is bad news. Yeah, it's a significant amount of money. But I think there are quite a lot of programs where, I don't know, you'll know better than me, James, but in peacetime, outside a pandemic, where a couple of months delay, a couple of months off the patent lifespan or whatever, that's worth a batch, you know, it's worth the possibility of having to spend £1 million or so on another batch. I think there's a lot of times it's actually, there is a business case for taking some more risk.
And I think that's where we've got partnerships where companies are willing to share risk. That's absolutely the case. I think what typically, that isn't a typical, you know, developer CDMO relationship. Typically, developers want things done and they don't want to take any risk, and they want it done as quickly as possible. So that puts all the sort of pressure on the CDMO, and you know, so in some relationships you can create that kind of mutual recognition and virtuous circle that supports a more rapid risk appetite, more rapid development program. And we've done work with partners where precisely that has we've taken on risk. We built, you know, with our first work towards the CAR T programs with Novartis, we built facilities at our risk, at our cost, to bring on facilities for a product that might not get approved. We didn't have that underwritten by Novartis. That was Oxford Biomedica that took that risk. But obviously we did it because we wanted to create a capability that would secure a long-term partnership with what was going to be, we hoped, the first approved CAR T therapy globally. And it, you know, with the benefit of hindsight, that's exactly what happened. But you know, in another alternative world, it might not have done. So you've got to think of it in that perspective. And every organization at the end of the day has to do the right thing by the shareholders. So if we're a publicly listed company, even a private company, they have to do what their owners want. In our case, it's our shareholders, and we can't knowingly, as leadership, do anything that might compromise the financial security of the business. And that is an underlying principle that I think is something that can be worked around through collaboration and through sort of shared risk management. But it does take a very collaborative partnership to be able to do it well.
I think one of the things I've learned is that, you know, if I'm asked to put together a project proposal for something, you know, I'm thinking back at, you know, academic research hat on here, you know, I will probably offer a proposal that is what the customer, in inverted commas, has asked for and is expecting, and is safe, you know. So yeah, good and fast and no compromise. And I'll say, well, actually, if you're willing to accept a bit of risk, or you're willing to, pay a little bit extra on this, well, we could do it in this time, or we could save you this much money. Here's an alternative. Might not be what you were looking for, but if you want us to do it this way, we can.
Yep. And we oftentimes with partnerships encourage adoption of projects that, particularly that, if you've got multiple parallel track activities, and some of those activities, like new cell line development, for instance, take a very long time. So stable cell line production, for instance, and encouraging partners to actually take the plunge on a development program for that. For some organizations, they think it's absolutely, sensible as anything, because the win on the positive, if it's successful, is huge. But others say, well, we'll decide to do that because at the moment we don't need it. We'll decide to do that later. And the latter approach is a safer but a longer time frame, and I guess it's getting that balance right, isn't it?
Yeah, and also balancing the needs of patients too, right? Certainly any drug development that's backed by patient advocacy will definitely see the value in time saved.
Yeah, indeed.
For sure.
So you asked me a question earlier, Jessica, but I can't remember what it was.
Yeah. It was a question that had come through. Oh yeah. Do you think the lentivector platform is the future or an alternative technology? And I'm not sure if it's referring to COVID vaccines or cell and gene therapy. So take it as you wish.
Yeah, well, I think first of all, I'll say, but I'm making the assumption that it isn't anything to do with vaccines. I think lentiviral vectors have many, many uses. And there are conceived projects which are effectively vaccine approaches. That said, I think they're typically lentiviral vectors are typically used in gene and cell therapy applications for complex programs, complex diseases where you've got to intervene in quite a difficult way. And I don't think lenti is going to be a challenging technology for a vaccine, because we need to make millions of doses, certainly for a pandemic, and right now, as of today, the doses that you need for the type of viral vectors that we make on our lenti platform don't generate millions of doses per batch. It's a simple fact, because of it's a different beast entirely. So I'm assuming it means related to gene and cell therapy specifically. I think the lentiviral vector platform has got a lot of potential for applications where it makes sense to adopt that. So for certain indications, certain approaches, things where ex vivo stem cells or T cells require permanent modification, such as the CAR Ts or the TCR-Ts, I think a lentiviral vector genetic modification has lots of pluses. I know others are trying to develop other non-viral approaches. For me, that other approach has got to be just as good, both from a cost perspective, a proven technology perspective, and a proven efficacy perspective. It's not enough to just be different from my perspective, because I think the safety database that's out there now in terms of genetically modified T cells in particular is pretty significant now. And I would also argue that, and I've always been I'm a virologist by background, and for me, using viral vectors is an obvious way of piggybacking on nature and taking a virus and basically harvesting the thing it does best, which is genetic delivery, and using that for your own intervention into a particular disease pathway. And there are plenty of examples in vivo gene therapy where other technologies can be good, can be conceivably much better in some cases than lenti. And it's also true that in certain indications in vivo, lenti has some significant conceived advantages. So for instance, for liver gene therapy, a lenti is a permanent genetic modification. So you can imagine, subject to proof of efficacy, proof of concept, and safety, you can treat patients whilst their liver, which is an organ that does turn over even in adulthood. You can treat them even in childhood and expect that therapy to be maintained into adulthood. That said, there are also plenty of other examples where AAV might make more sense, or other gene therapy vector approaches might make more sense. So I think I made the point earlier, we have a core expertise on Lenti as a business. That's what we were founded on. I think what we've found and proven now is that having competence in manufacturing and developing Lenti, both in the manufacturing processes and analytics and the core technologies, does have benefits in terms of being able to be, the skill set, the equipment, the facilities, unit operations, they can be readily applied to other platforms, such as Adeno, we've already proven, and AAV. So from my perspective, we are, as a company, less specifically focused on Lenti. We had a lot of work to do on Lenti, being the first commercial manufacturer. That was a huge piece of work for us as a business, and really that had to be our focus. But right now, I think we've come to the stage where we've got sufficient bandwidth and sufficient broad experience to sort of really engage in working on other technologies, and other viral vectors being a core example, that utilize those skills and our competence, and the fact that we are an approved commercial manufacturer for a product that is now sold globally, and now we're a commercially approved manufacturer for 2 products that are sold globally. That in itself is a sort of a line in the sand, which proves that we are competent and we can do this effectively at scale and to the standards that the agencies around the world and the commercial manufacturers of big pharma expect.
Yeah, so a question, we've talked about how this whole experience has, there's been a lot of lessons learned on how to maybe manufacture the, ChAdOx for COVID or for other diseases. How has the experience helped Oxford Biomedica think about cell and gene therapy differently, maybe?
Yeah, so I think one thing that is important, I think, from the Adeno experience has been, historically we haven't there are no cell and gene therapy products that require as much product, right? So what we found, and we already knew this, but working in a cell and gene arena, there aren't that many products and projects that require basically back-to-back repeat batch manufacturing, and I think repeating things is a very good way of utilizing your capacity efficiently. That allows us, and we make sure that we are doing a responsible manufacturing for a product that's for the greater good. And I think it's been widely publicized that AZ is taking a low cost approach to this product. I think that's important, but it is a business, and it's profitable to the extent that we can reinvest and allow us to further, move into other territories and into other products. So I think having that sort of constant revenue stream, if you like, is really helpful from a business security perspective. And it also allows us to take on more projects, and sometimes more risky projects where they may, the proof of concept might not be anywhere close to likely to happen in any time soon. It's still worth doing because the long gain is there to come in the future years.
Yeah, that makes a lot of sense too. I think so. Another question that's come through is on a topic we haven't discussed yet, which I think is an important part of the conversation, which is that of the regulatory environment. That during the course of last year, as this product was rapidly moving through development. So how were the interactions with the UK regulatory agencies and globally through this process?
Do you want to start that? Yeah.
I'll start on that. I mean, so the university has had a strong dialogue with MHRA, the UK regulators, over many years. We've been regular users of the MHRA's Innovation Office service when we've been wanting to do something a bit different from standard practice, and we've found we've had excellent scientific advice from them, and very much valued the fact that they'll take a science led approach and very much consider patient benefit as well as process. So we found that from the beginning an essential enabling part of our COVID vaccine development program. Clinical trial applications were turned around within 24 hours. Rolling submission of everything was standard. and throughout the agency maintained rigor, but also was at pains, I think, to make sure that that didn't mean distance in terms of availability to discuss what needed to be discussed.
Yeah, and I completely agree. I think the MHRA, the UK MHRA, is an organisation that has a lot of interest in innovative approaches. They even have an office called the Innovation Office, and their approach is, absolutely focused on safety, but they were actively engaged in what could they do to support rapid build-up of manufacturing capability and so on. So, I've already mentioned our Oxbox facility had been handed over for us to start the qualification and commissioning and ultimately the approval for the MHRA GMP license early in 2020. So when Sandy and I had our first conversation, we didn't have an MHRA license on that facility. We were planning one already for our Lenti programs and had it in the schedule, and had already got to the point where we'd submitted, I think, from around that point. So we knew when we were going to get inspected, but we weren't planning on bringing all 4 facilities on as quickly as possible. 3 of them with completely new process and new equipment and lots of new people. So that did require a lot of dialogue. And it required the MHRA to be, basically willing and able to provide resource to come in and do a GMP inspection. Their standards were no different from normal. We've been a GMP accredited organisation for many years and have had a lot of experience in MHRA inspections over the years. So, the level of rigor that they went into was really no different. They just put more kind of rapid energy into it, if you like. So, and actually, they also, given it was a COVID project, they overtly prioritized that activity. And, to the extent where some other activities that weren't COVID, including some we were doing, we knew would be pushed down the priority list. So, and that was, I guess, an expected, not unexpected, I suppose, outcome of the situation. But we then made sure we were closely aligned with the MHRA, and we were telling them very, very regularly where we were with qualification activities. And I guess probably the best example of how it worked was we had a production schedule, and our production schedule for our 3 suites for vaccine was a very complex schedule with lots of overlapping shift patterns and personnel management. We developed that before we had approval on the GMP 7 and 8. So we had to know that the MHRA would be able to come in and inspect the iterative difference, if you like, between GMP 7 and GMP 6, and then subsequently GMP 8 and GMP 7 and 6, to demonstrate that we were compliant. But they could only do that when we were ready. So,
right.
So we had to coordinate it really, really carefully, and they were absolutely amazing. They basically worked to our schedule. We said we're ready within a day or 2. They were able to then conduct their inspection and give us the approval, and then within a day or 2 of that, we'd started manufacturing in each of those suites.
Incredible.
I think there's something quite interesting for me about the culture of the organization, and I know nothing about how this is created, but the feeling I have as a not that expert outside observer is that there is a culture within the MHRA of individuals absolutely in the organization absolutely being accountable for and having a mission for patient safety, but they also are, as I said, placing value on patient benefit and placing, to an extent, value on industrial innovation as well. And I think how to create that, not just in a regulatory environment, but perhaps also in quality teams as well, is in other contexts beyond COVID, while it's not an easy thing because it's requiring individuals to, the safe thing for an individual in a position of responsibility like that is to defer to process. So how to create a culture which enables people to think and value a number of different outcomes as well as process, Really important question.
Yeah, I agree.
I think, as you started in the beginning of the conversation about the regulators, at the end of the day, they're scientists. You'd like to think that, with sound science, they will be able to make decisions and adapt, as long as the data is presented and the stance is reasonably described.
Yeah, I think the quality of the scientists going in is clearly important, and the MHRA is staffed by good scientists. It's had experience built up over many years. And, not every country, this is an important thing. Part of the debate about vaccine equity of vaccine access is actually about improving regulatory capacity in other countries around the world. We're in a fortunate position here to have that. And I really hope that the message gets through loud and clear to government that investing in having good people in that environment is probably one of the smartest investments for UK public health and the economy that they can possibly make.
I think we're near the end of the hour, and I feel like that was a nice, strong statement to end on. So I don't know, James, if you have a final comment, or shall we?
No, I think it's a very interesting topic, and I think we could probably talk about this for hours, but we won't dwell. I think we've covered off a lot of the sort of questions that I think that have been raised. So yeah, been a pleasure talking to you all.
Yeah, thank you everyone for joining us today and listening and participating and submitting your questions. We tried to answer most of the ones that came through, and it's been a pleasure talking to you.
Thank you all.
Thanks.
Yeah, unfortunately, that is all that we have time for today. So I do apologise if we didn't get around to addressing your questions. Before we wrap up, I would just like to say thank you to Jessica, James and Sandy, not only for their insights, but for joining us today. As always, you can find this webinar along with all of our others via the Facilitate website, www.facilitate.com. You can also still register to join us all in Miami via the same website. But until next time, thank you for joining us today and goodbye.